Hi, good evening. In this session, I am going to discuss jet scheme, which is also called as non-cyclic photophosphorylation. The name itself indicates non-cyclic photophosphorylation. Phosphorylation, formation of ATP. Photophosphorylation, formation of ATP in the presence of sunlight. Non-cyclic photophosphorylation, formation of ATPs in the presence of sunlight during the non-cyclic movement of electrons is said to be non-cyclic photophosphorylation, and the same is called as jet scheme. So the jet scheme name is given on the basis of the pathway of the electrons. Here jet scheme includes both the pigment systems or photosystems, you can say. Photosystem 1, photosystem 2. Whereas in the case of cyclic photophosphorylation, only PS1 involves. But in the case of jet scheme or non-cyclic photophosphorylation, both the PS1 and PS2 involves. So by this, you can guess where does it occur. It occurs in granathylakoids because the stromathylakoids, they consist of only PS1, whereas the granathylakoids consist of both PS1 and PS2. As PS1 and PS2 are located on the granathylakoids, the jet scheme will occur on the granathylakoids. So here there are two photosystems like PS1, PS2. Here light harvesting complexes are formed. This group of pigments together constitute light harvesting complex. Actually here the molecules absorb the light in the form of photons. You know, the sun emits the radiant energy which is absorbed by these pigments. Radiant energy comes in the form of quanta or photons. Photons are absorbed by the pigment molecules, and they will transfer that photons to the reaction center. In the case of PS1, the reaction center is a special chlorophyll molecule which is called P700. Actually, that 700 or this 680, what do they represent? They represent the absorption maxima, the maximum wavelength of the light which can be absorbed by that pigment system. So P700 means they can absorb the light of more than 680 and the maximum is 700. So they can absorb the light of 700 nanometers. That is the maxima, absorption maxima. So they can absorb above the 680, below the 700, and the maxima is 700. In each light harvesting complex, many molecules are there like chlorophyll A, chlorophyll B, xanthophylls, carotenes, all are present. Here the xanthophylls and carotenes together constitute carotenoids. Actually, these act as accessory pigments or the shield pigments which absorb high wavelength of light. By absorbing the high wavelength of light, they prevent the chlorophyll molecules from the photo bleaching and the photo oxidation or solarization. At the high wavelength of light, chlorophyll molecules undergo solarization, chemically destructed. If they are chemically destructed, they cannot trap the sunlight or radiant energy. So here some accessory pigments are there known as carotenoids. They absorb maximum wavelength of light. By absorbing the maximum wavelength of light, they prevent the chlorophyll molecules from the photo bleaching process. So that they are called shield pigments, otherwise accessory pigments. However, they also part of the antenna. Here the light absorbed by the pigments will be transferred from one pigment molecule to another pigment molecule like this. This transfer of energy from one pigment molecule to another pigment molecule of the light harvesting complex is called as resonance transfer. And finally, the light will be transferred to a special chlorophyll molecule which acts as a reaction center. Now, the reaction center gets excited. That is a later story. So here there are two types of photosystems or pigment systems known as PS1, PS2. PS1's reaction center is P700, that of the PS2 is P680. Both 700 and 680, they represent the absorption maxima of that LHCs, light harvesting complexes. So here, first this PS2, the reaction center P680 gets excited because the photons are absorbed by the light harvesting complex, and that is transferred through various pigment molecules to the reaction center through the resonance transfer, and finally that is transferred to the reaction center P680. Then the P680 gets excited. In that excited state, the electrons are jumped into the orbit which is present farther from the atomic nucleus. And in that excited state, the reaction center is denoted with a star mark above that, like P680 with this symbol. Now, in that excited state, two electrons they jump into outer orbit now. From that, an electron carrier picks up that electrons. Actually, electron transport chain is present. Electron transport chain is nothing but a series of intermediate electron carriers over which the excited electrons are transferred in downhill. In this downhill journey, at every step, some amount of energy is released. That energy is utilized to develop the electrochemical photon gradient, otherwise proton concentration gradient in between the stroma and the lumen of the thylakoid. Now, the electrons which get excited from the reaction center P680, they transfer to the PO15. PO15 is a electron carrier. This is the first electron acceptor, and the PO15 is nothing but a chlorophyll molecule without magnesium. Chlorophyll molecule without magnesium is colorless. That colorless chlorophyll molecule without magnesium is called as PO15. In the place of magnesium, two protons are present in the PO15. This acts as a first electron acceptor that accepts the electrons from PS2. Here, each and every electron carrier which is present in the electron transport chain gets reduced and subsequently oxidized. When it receives electrons, gets reduced. When it transfers electrons to the adjacent electron carrier, gets oxidized. So that depends on their redox potential, which can be measured in volts or millivolts. It is a measure of tendency to acquire the electrons and thereby gets reduced. Here now PO15, it transfers electrons to plastoquinone, which is present on the outer surface of the thylakoid membrane. Here, plastoquinone transfers electrons to the cytochrome B6F complex. During the transfer of the electrons to the cytochrome B6F complex, their quinone cycle takes place. Quinone, plastoquinone first initially gets reduced to semi-plastoquinone or plastosemiquinone. Later, further it gets protons from the stroma and completely reduced to PQH2, plastoquinol. And finally, the electrons are transferred to the adjacent electron carrier and the protons are pumped into the lumen, like this, into the lumen. From the stroma, protons are removed and pumped into the lumen by the plastoquinone. The role of plastoquinone is to transfer electrons from PO15 to cytochrome B6F complex, and that is coupled with the pumping of protons from the stroma into lumen. Now, the electrons are finally transferred to the plastocyanin, which is a mobile electron carrier. By this time, simultaneously, here in PS1 also the pigment system, it absorbs the photons and that is transferred to the reaction center P700, and the reaction center P700, which is nothing but a chlorophyll molecule, gets excited. In that excited state, again the electrons are ejected. The ejected electrons are transferred to the ferrous sulfur protein. Later, the electrons are transferred to the ferredoxin, and from there the electrons are finally transferred to the NADP plus, which is an oxidized state under influence of FNR, ferredoxin NADP reductase enzyme. So that here it gets reduced to NADP minus. Later it remove the protons from the stroma and thereby get reduced to NADPH plus H plus. Like this, the electrons which are ejected from the reaction center P700 finally transfer to the NADP. They are not cycled back to the P700. Likewise, the electrons which are released from the P680 reaction center also transfer to the PS1. Here the electrons they are removed and finally added to the NADP. Here electron deficiency created. That electron deficiency is fulfilled by the electrons from P680. So P680 donates electrons to fulfill the electron deficiency in P700. As a result, here electron deficiency is created in the PS2 because the electrons are transferred to PS1. Electrons of the PS1 are transferred to NADP. So finally their NADP reduced to NADPH2, and here deficiency is fulfilled by P680, and here deficiency is created, electron deficiency, electron lacuna is created. Now, to compensate this electron loss, water molecule undergo photolysis. So, what is photolysis? Lysis of the water in the presence of sunlight is said to be photolysis. This is connected to OEC, oxygen evolving complex, which is present on the inner surface of the thylakoid membrane that consists of manganese ions and calcium ions and chlorine ions. With the help of all that ions, here water molecules undergo photolysis. When they undergo photolysis, for example, if you take one water molecule, it gives rise to half molecule of O2, 2 electrons, 2 protons. Likewise, if you take 2 water molecules, they give 4 electrons, 4 protons and 1 molecule of oxygen. The protons are released into the lumen because this photolysis occurs inside the lumen. So protons are released into the lumen where the electrons are transferred to the reaction center P680, because in the reaction center P680 electron deficiency is there because that was created due to the transfer of electrons from P680 to P700. Now this lacuna is filled by the photolysis. Photolysis not only results in electrons but also results in protons. Electrons are transferred to P680, protons get accumulated there itself in the lumen of the thylakoid. You see, now here during the transfer of electrons from plastoquinone to cytochrome B6F complex, some protons are pumped into the lumen. Even this photolysis also results in the accumulation of the protons. As a result, and here NADP remove the protons from the stroma. All these activities decrease the proton concentration in the stroma and increase the proton concentration in the lumen. So photolysis increases proton concentration in the lumen, whereas the transfer of electrons by the transfer of electrons from plastoquinone to cytochrome B6F complex also, at that step also protons are pumped. This pumping of protons by plastoquinone decreases proton concentration in the stroma, increases proton concentration in the lumen. And this NADP, it removes the protons from the stroma and decreases proton concentration in the stroma. Because of all these activities, a concentration gradient is established in between the stroma and this lumen. When this concentration gradient, it develops that membrane potential, and this membrane potential and this electron, this proton concentration gradient together constitute proton motive force or electrochemical gradient. When it is broken, then ADPs are formed through the chemiosmosis. So here, in non-cyclic photophosphorylation, electrons that are removed from the P680 are not cycled back to the P680, and electrons that are removed from the P700 are not cycled back to the P700. The pathway of electrons is zigzag, so it is called jet scheme. So here NADPH2 generated, ATPs are generated, oxygen is evolved, and both the pigment system 1, pigment system 2 or photosystem 1, photosystem 2 involved. Okay, here how many quanta of the light required for the occurrence of this jet scheme? We need 8 quanta, because for the photolysis of one water molecule we need 4 quanta, because for the photolysis of one water molecule 2 pairs of electrons should get ejected. So 1 pair of electrons at the P680 and 1 pair of electrons at the P700. So here, for the ejection of 1 pair of electrons, 2 quanta are required to exit the P680. Likewise, for the ejection of 2 electrons at P700 also, we require 2 quanta to exit the P700. So for the photolysis of one water molecule, if photolysis of one water molecule takes place, half molecule of oxygen is produced. For the photolysis of one water molecule, we need 4 quanta. 2 quanta at P680 means PS2 and 2 quanta at P700 means PS1. If one is photolysed, half molecule of oxygen is released. To get 1 molecule of oxygen, we need 2 water molecules. So for the photolysis of 2 water molecules, we need 8 quanta. For 1 water molecule, 4 quanta. For 2 water molecule, 2 into 4, 8 quanta. Of them, 4 quanta they work at the P680 and the 4 quanta they are needed at P700. So this amount of light or the amount of photons or quanta which are required to evolve one oxygen is called quantum yielding. Quantum requirement. Quantum requirement is the light quanta required to evolve the one molecule of oxygen is called quantum requirement. Quantum requirement is equal to 8. Quantum requirement is equal to 8. Of these 8 quanta, 4 quanta are required by the P680 and the 4 quanta are required by P700. Here, the amount of light that is required to evolve one oxygen molecule is called quantum requirement. Why oxygen is evolved? If photolysis takes place. So for the photolysis of one water molecule, 4 quanta, 2 quanta at PS1, 2 quanta at PS2. For the photolysis of 2 water molecules, 8 quanta, 4 quanta at PS1, 4 quanta at PS2. So each water molecule it releases half molecule of oxygen or one atom. So 2 water molecules can produce 1 molecule of oxygen. Then what it is quantum yielding? So quantum yielding is nothing but the amount of oxygen produced for the absorption of 1 quantum. The amount of oxygen which is produced when 1 quantum is absorbed. So quantum yielding is nothing but 1 by 8, that is equal to 0.125, otherwise 12.5 percentage. So quantum requirement, quantum yielding. Quantum requirement means the amount of quanta required to evolve the one oxygen. Quantum yielding means the amount of oxygen which is evolved on the absorption of 1 quantum, that equal to 1 by 8. Quantum yielding equal to 1 by 8. Quantum requirement is equal to 8 quanta. So like this, non-cyclic photophosphorylation takes place, which is present in the plants.